Everybody, the topic for today is drug metabolism, and this is under Module 5. If you will check, may tatlo pa tayong modules remaining to be able for us to finish midterm grading period. So our goal is to finish drug metabolism until tomorrow. Hopefully matapos natin siya bukas, earlier than what is expected. Technically, drug metabolism is divided into 3 lessons. But I've decided to have 5 parts para matapos natin overall ang drug metabolism. The following are the contents of this module. Number 1, I will discuss introduction and also importance of drug metabolism, in which our goal here is for you to understand what metabolism is all about and its importance, especially sa mga gamot. And after that, we will also tackle the different factors affecting drug metabolism. And then we shall proceed on the general pathways involving metabolism of drugs, in which we will discuss chemical reactions that are categorized under phase 1 and phase 2. Okay? So let's start by discussing introduction to drug metabolism. By the way, let me emphasize that the topic metabolism here is quite different with the one that you discussed in biochemistry before. Okay, iba siya. Dahil dito nakafocus tayo sa drugs, not biomolecules. All right? Regarding drug metabolism as an introduction, remember that drugs are important substances responsible for pharmacological actions. The reason why we take drugs is for us to feel the effect and para gumaling tayo sa sakit. Now, it should be realized that drugs effect should not be perpetual. When we say perpetual, hindi dapat siya habang buhay, hindi dapat siya long term, or hindi dapat siya pangmatagalan. Otherwise, drugs could already produce toxic effects. That's why it is important for drugs to be excreted. Ay importante din na ang gamot ay ma-excrete sa ating katawan. Now, for drugs to be excreted, there has to be a prerequisite or major requirement. That is, a drug should undergo the process called metabolism. That's why it is very important for us to understand what metabolism is all about. Can you follow? So let's continue. Synbiotics require to undergo drug metabolism for them to become hydrophilic metabolites. When we say hydrophilic metabolites, as you remember during our discussion on Module 1, this means to say that drugs or medications are already ionized. They are already ionized, and also they are said to be polar. So I repeat, when they are hydrophilic, this also implies that they are ionized and also polar. This would only happen if drugs underwent metabolism or drug metabolism. Hence, ang metabolism is also known as biotransformation. Again, that's why metabolism is also known as biotransformation. Ibig sabihin ng biotransformation, we are converting, we are transforming, ay ginagawa natin ang gamot na hydrophilic, na ionized at polar. Tama ba tayo na kapag ang gamot ay naging hydrophilic, ionized o polar, mafe-favor na ang excretion process? Very good. That's why ma-facilitate na ang paglabas ng gamot, kung ano man ang mga routes of excretion na pwede or applicable sa kanya. Next, so continue natin. In drug metabolism, yes, magiging hydrophilic siya, ionized at polar, and metabolism would only happen because of biological catalysts called enzymes. Again, we have the biological catalysts called enzymes. These are the ones responsible for different chemical reactions in the body, particularly in hastening them. Pag sinabing hastening, pinabibilis ang chemical reaction, forming products. Now, highly concentrated ang enzymes in the hepatic tissues or hepatic cells. That's why we also deemed before that the major organ for metabolism is the liver. Again, the major metabolizing organ is the liver. However, please bear in mind also that hindi exclusive sa liver lang ang mga enzymes for metabolism. There are also other tissues or organs in the body capable of metabolism. Basically kasi meron din sila mga metabolizing enzymes. So ang tawag doon, extrahepatic enzymes. Later, we shall cite examples of organs or tissues containing or possessing these extrahepatic enzymes. Next, so let's continue. Now, the major importance of drug metabolism is to identify or to determine or even to assess drug safety. Via drug metabolism, we can understand the different pharmacological and toxicological activities of drugs. In Tagalog, importante ang metabolism upang malaman natin kung ang gamot ba ay safe o kaya magiging toxic na siya. That's why related nga dito ang pharmacological actions and toxicological activities. For example, sa pharmacological activity, through metabolism, we can identify if the drug is already inactive, ready for excretion, or in some cases, pwede nating malaman na ang gamot ay active through metabolism. Again, pwede nating malaman through metabolism if drugs are already inactive, that means hindi na sila capable of their actions, o kaya via metabolism, we can also identify if drugs are active or activated. Ano yung example case natin na na through metabolism ang gamot ay na-activate? That is in the case of prodrugs. Can you still recall prodrugs, guys? Very good. That's why important siya. And also inactive, ito yung common fate na mga drugs. Mamaya palalalimin pa natin siya. Moving on, what about toxicological activities? Just to emphasize toxicological activities, tandaan, generally speaking as well, the major goal of metabolism, aside from making the drug inactive, as much as possible, dapat maging non-toxic siya. However, there are cases wherein because of metabolism, drugs became toxic. Kaya pwede natin ma-assess ang toxicological profile ng gamot through metabolism identification or determination. For example, very common dito, si acetaminophen, di ba? Si acetaminophen. Bakit ang acetaminophen ay well known in causing hepatotoxicity? That's because its toxic metabolite ay yung toxic metabolite niya ay nagkukos ng liver damage. So bakit nagkaroon ng toxic metabolite? Kasi na-metabolize siya. That's why we need to study metabolism for us to realize the toxic profile of acetaminophen and other related drugs. Later, magbibigay tayo ng instance or may specific part ata ako ng lecture about acetaminophen. So stay tuned na lang muna. But so far, nagigets ba ako dun sa importance, general importance? Very good. Okay, next. Yung susunod na slide na na dapat nasa notes nyo, hindi ko muna siya ipapakita sa ngayon because there is another slide na dedicated for that part. Yung mga drug-drug interactions, drug food, can you follow? Meron akong other part kung saan ko siya mamimension, kaya skip na lang natin siya. Gets ba ako, guys? Okay, so punta tayo sa part na to. Ready na kayo sa part na to sa important note? Ready? Very good. So let's proceed to this important note. Remember that the general goal of metabolism is to make the drug hydrophilic. However, there are medications that do not need to be metabolized already. In Tagalog, may mga gamot na hindi na kailangan mag-undergo ng drug biotransformation. Why? Because there are drugs that in the first place are already hydrophilic. Again, because there are drugs that are in the first place hydrophilic. And when they are hydrophilic, the major route of transport para makapasok ang mga gamot na to sa cells ay hindi na passive diffusion. Rather, it's via paracellular transport. When we say paracellular transport, this is applicable, I repeat, sa mga hydrophilic drugs. The process of paracellular transport means that drugs will enter the cell through the presence of cellular pores. Again, that's because of the presence ng mga cellular pores. When we say pores, this can be seen beside each cell or cell membrane. Halimbawa, just for you to understand, halimbawa, this box represent one cell, okay, or one cell membrane. And then we have another, okay, we have another, and then we have another. Halimbawa lang, hypothetical lang siya. Now, please note that hindi literal na attach sa isa't isa ang mga cells. There are gap junctions or pores kung tawagin. And these pores are butas that would serve as opportunity for hydrophilic drugs to enter. Kaya hydrophilic drugs enter through paracellular transport. This is in contrary to some drugs that are lipophilic kasi ang lipophilic ay dumadaan mismo sa cell membrane via diffusion. Okay, yun yung difference. So nagets nyo guys, yung ibig ko sabihin? All right, going back with our discussion, since these are in the first place hydrophilic, they don't need to be metabolized. They do not undergo metabolism. And ito yung mga nababasa nyo sa mga references na excreted unchanged. Pag may nabasa kayo sa literature o kaya sa mga drug insert na ganitong percent, halimbawa lang 20% of the drug is excreted unchanged. Ibig sabihin, hydrophilic na yon and they are resistant to metabolism or biotransformation. So that's an important note about drug metabolism. Kaya when the time comes na makabasa kayo ng mga ganitong information, somehow may idea kayo na kapag unchanged, nag-shortcut kumbaga, bypass na yung metabolism, diretso excretion na kasi hydrophilic na yung gamot in the first place. Gets? Naintindihan? O yung iba, kumusta kayo dyan? Palalim ng palalim yung discussion natin. So yung ano muna, general concept muna. Good. Next stop. Second part na tayo ng ating outline. Let's proceed to the different factors affecting drug metabolism. Listen carefully. Key concept about this portion. Drug metabolism is not absolute. When we say not absolute, hindi same ang fate ng gamot sa lahat ng tao or lahat ng living organisms. There are different factors that could affect drug metabolism, resulting to certain variations. In that case, let's discuss each factor. Paki-ready yung ebook kasi ito yung part na magre-refer kayo sa mga page numbers. First, we have genetic factors. Of course, genes do dictate certain characteristics and also synthesis of enzymes. What I mean to say is because of the genetic factor, maaaring mag-vary yung mga enzymes na napoproduce sa ating katawan and also that could result to variation of their activities. And the field related to the study of the relationship between the genes and also patient response to a certain drug treatment is called pharmacogenomics. Kaya genes, very important factor to consider. All throughout our discussion, meron kayong mangilan-ngilang mga examples na may encounter that you can relate to genetic factors. So by the time na umabot tayo doon or mag-proceed tayo doon, I will always remind you about genetic factor. Okay? For example, Asians, Americans, Caucasians, Black, Africans, etc. So, regarding this part, let me give you a representative case para mas ma-imagine nyo kung ano yung pinopoint out natin sa genetic factor. This has something to do with the metabolism of ethanol. Okay? Again, this has something to do with the metabolism of ethanol. Are you familiar to ethanol? Good. Ethanol is also alcohol in pharmacy. So ang ethanol, pag nakapasok siya sa ating katawan, meron siyang metabolic fate. At this point, let us lecture or discuss the metabolic fate of ethanol once it enters the body. You have to note that ethanol in the first place as an organic compound, is an alcohol. And the fate of an alcohol sa organic chemistry ay oxidation. Okay, oxidation. Lalong-lalo na ang ethanol ay isang primary alcohol. Ay lalong-lalo na isa itong primary alcohol. With that being said, let's talk about the fate of ethanol once it undergoes oxidation. So first, as an alcohol, it will undergo it will undergo its first oxidative reaction, which is being catalyzed by the enzyme alcohol dehydrogenase. Okay, this is being catalyzed by the enzyme alcohol dehydrogenase that would lead in the formation of acetaldehyde. Again, that causes formation of the intermediate product acetaldehyde. Now, what's with acetaldehyde? Apparently, acetaldehyde is an aldehyde. Okay? Aldehyde siyang organic compound. And acetaldehyde is considered toxic in the body, especially pag siya ay nag-accumulate because it can it can cause nausea and vomiting. It can cause nausea and vomiting. And also, it can cause tachycardia. Familiar ba sa tachycardia, guys? I bet you are familiar with tachycardia, no? Okay, tachycardia and also redness. Okay, it can cause redness. So these are some of the effects caused by acetaldehyde. So if you are curious about acetaldehyde's other effects, you can check Google. Okay? Ito lang yung mga common effects caused by acetaldehyde. Kasi nga toxic siya, especially when accumulated. Now, inside the body, pwede na natin continue by the way. Very good. Now, inside our body, metabolism of alcohol or ethanol does not end sa acetaldehyde. Ang acetaldehyde will further undergo oxidation which is held by the enzyme aldehyde dehydrogenase naman. This is being held by the enzyme aldehyde dehydrogenase leading in the formation of acetic acid, ay leading in the formation of acetic acid. Relatively, ang acetic acid is non-toxic. Again, relatively, acetic acid is non-toxic that is readily excreted. Again, this is non-toxic and readily excreted. So overall, this is the metabolic fate of alcohol kapag siya ay nag-undergo ng oxidation. So ang takeaway natin dito is, again, makiniga, ito ang takeaway natin. Number one, ang acetic acid ang ultimate product ng ethanol metabolism and this is non-toxic. Number two, nangyayari ito because of metabolizing enzymes alcohol and aldehyde dehydrogenases. Are you done copying? Type done if you're finished copying. Okay, makinig mabuti. Mag-focus tayo ngayon sa mga enzymes that were written. Specifically kay aldehyde dehydrogenase. Again, mag-focus tayo kay aldehyde dehydrogenase. Genetically speaking, again makinig, genetically speaking, aldehyde dehydrogenase expression is not the same in all races. Meron tayong mga specific races wherein ang aldehyde dehydrogenase ay poorly expressed. Pag sinabing poorly expressed, mababa ito sa kanila. At given ito sa mga tao like Chinese, Chinese o kaya Japanese, sa mga Japanese. I repeat, aldehyde dehydrogenase is poorly expressed to certain races like Chinese and also Japanese. So with that being said, makinig, with that being said, kung mababa ang expression ng aldehyde dehydrogenase at sa mga Chinese at Japanese, do you think may chance mag-accumulate ang acetaldehyde sa kanila? Yes or no? Yes. So that can cause accumulation of acetaldehyde in these patients. Kaya ang tendency, sila ay namumula, nagtatakicardia, nagsusuka. So anong implication niyan? Yung mga Chinese at Japanese, kapag nom-nom sila ng nom-nom, umiinom sila ng marami, mabilis silang mamula. Ang tawag doon ay alcohol intolerance. Okay? Ang tawag doon ay alcohol intolerance. Na-gets? Hmm? Naintindihan? Hmm? Okay, so ang tawag doon ay alcohol intolerance. Kaya kayo, kapag umiinom kayo at napansin nyong namumula kayo, pwede nyo sabihin, poorly express ang aking aldehyde dehydrogenase. May lahi akong Chinese. Baka may lahi akong Japanese. So it comes with genes. It comes with genes. So poorly express ang aldehyde dehydrogenase. So, ang tawag doon, alcohol intolerance. At least meron kayong mechanistic approach para mas maintindihan ang alcohol intolerance and also i-explain nyo sila sa mga kainuman nyo o kainuman nyo pag umiinom kayo. But kidding aside, did you get this part? Okay, next. Punta tayo sa second factor. Pwede na sa second factor, physiologic factors. So under physiologic factors, of course, it depends on someone's physiology kung ano ang magiging response niya sa metabolism of drugs. So these are the following. Some of them were indicated in your ebook pero some wala. Kaya please be guided accordingly na lang. So for instance, we have age. Explanation on age could be found on page 48. So it's your task to check page 48 and read it. May chance na lumabas siya sa task o kaya exam, kaya please pakibasa. So ang guide nyo dito lang, again, ang guide nyo dito, alamin nyo kung ano ang common response or sample response ng mga pediatric patients, ng mga pediatric patients and also geriatric patients and also adults. Ano yung mga variations sila? Okay, so pedia and also geriatrics. Next, we also have pregnancy. Of course, there is a different approach or response ng mga pregnant women sa drug metabolism. Ay, syempre, mas sensitive sila. Next, we have hormones. Hormones because hormones of a man differ from the hormones that a woman has. Kaya nagva-vary din siya. And in relation to hormones, we have gender, of course. Paki-check yung mga sample responses, yung mga studies conducted about responses of male and also females sa mga certain medications na sinite sa page 48. Can you see that, by the way? Am I correct on the page number? Chinek nyo ba? O, i-check nyo ha. O, ano nyo na yan? Homework nyo na siya. Reading assignment nyo na siya. Maaari lumabas sa tasks o kaya sa exam. Also, we have changes in intestinal flora because ang intestinal flora they have metabolizing enzymes or metabolic activity as well. Meron silang metabolic activity and also some disease states. There are disease states that would cause enhanced activity of enzymes, meron namang reduced activity. It depends on the situation and of course, nutritional status. So overall, these are the different physiological factors affecting metabolism. Next, what about pharmacodynamic and pharmacokinetic factors? This could be found on page 49. Regarding the specific example, sa phase 2 ko ito madidiscuss. Actually, ito yung last slide nung binigay ko sa inyo sa notes. Ay, last slide. Madidiscuss natin yan later, so do not worry. Doon muna tayo sa general factor under dynamic and kinetic. Remember, during our Module 1 discussion, ang dynamic, it has something to do with the effects or yung molecular actions by drugs. Pag kinetics naman, it's the ADME. Again, these factors could affect drug metabolism such as dose, or kaya frequency of administration and also route of administration. Sa dose, ano ang example natin sa dose? Of course, the higher the dose, the greater the extent of metabolism kasi marami ang kailangang ma-metabolize. As simple as that. Okay? Frequency, the more frequent, the greater the frequency of metabolism also kasi mas madalas ang intake, mas madalas ang requirement for excretion. That's why greater frequency of metabolism. Route of administration. Ang point sa route of administration, there are cases that ang gamot ay less ang requirement for metabolism o kaya more. Halimbawa, if the route of administration is intravenous, less likely siyang mag-undergo ng high extent of metabolism kasi nababypass, for example, ang first pass effect. Something like that. Kaya somehow, yung discussion natin sa part na to ay prerequisite yung Module 1 topic kasi ina-apply natin right here, right now. So route of administration, in some cases, we can bypass some metabolic reactions like first pass effect in the case of intravenous administration. Also, tissue distribution, protein binding, these are the factors affecting metabolism, pharmacodynamic and kinetically speaking. Okay? Next, punta tayo sa number 4, environmental factors. So under environmental factors, just like sa mga medications, there could also be interaction with environmental substances. So you can read page 49 under environmental factors, kung ano yung naka-indicate doon. It's part of your reading assignment also. And then finally, dito tayo mag-focus sa number 5 kasi I think this requires explanation. Number 5, ito yung tinalunan kong slide kanina. Yung part na sabi ko babalikan natin. So ito na yon sa number 5. Let us proceed to drug-drug and also drug food and finally drug supplement interactions. Marami ang mga possible drug-drug, drug food and drug supplement interactions, but we shall specify here interactions that could alter metabolism. Alteration, ang tawag doon everybody, alteration of metabolism. Again, alteration of drug metabolism. So through this alteration process, we can change, we can modify the potential activity of drugs. So meron tayong dalawang cases. By the way, nakakasunod ba? Nakakasunod? Parang napabilis ata ako. Anyway, nasa book naman, open yung 49 na, pupunta tayo dyan later. So going back, we have alteration of metabolism. Meron tayong dalawa. We have two possible situations that are categorized under alteration of metabolism. Number 1 is enzyme induction. Second, we have enzyme inhibition. I repeat, we have enzyme induction. And second, we have enzyme inhibition. Actually, na-discuss na natin ito no Module 1 pero may dadagdag lang ako mga concepts. First, for enzyme induction, remember, pag enzyme induction, can you tell me overall what happens to the rate and extent of metabolism? Enhanced or reduced? Pag enzyme induction. Nice. So that causes enhanced rate and extent of metabolism. And then, on the contrary, kapag enzyme inhibition, reduce metabolism. Alam nyo na yan, na-discuss na yan. Ngayon, dagdagan natin, as I've said. In enzyme induction or enhanced metabolism, of course, this will happen if there is a cause, kapag may dahilan. Maaaring ma-enhance ang metabolism ng gamot kasi meron tayong mga meron tayong suspect kung bakit nagkakaroon ng enhanced metabolism than the usual involving a drug. Ang tawag natin sa suspect para magkaroon ng enhanced activity or enhanced metabolism, mga enzyme inducers. Again, ang tawag doon ay mga enzyme inducers. Next, kapag naman enzyme inhibition, ang ating suspect ay enzyme inhibitors. Apparently, enzyme inhibitors. I repeat, we have enzyme inducers as suspects para magkaroon ng enzyme induction involving a drug and also enzyme inhibitors as suspects para mangyari ang enzyme inhibition. Now, saan nyo makikita ang mga common examples ng enzyme inducers and inhibitors that could be found on your ebook at page 49? Kindly open your ebook and proceed on that page 49. Can you see the table that lists down the enzyme inducers and inhibitors? Yung metabolism, inhibiting, and inducing drugs. Can you see that? Yes, okay. Nice. So makikita nyo dyan ang metabolism inhibitors and metabolism inducers. So ito yung mga suspects. Again, ito yung mga suspects para magkaroon ng alteration ng metabolism. Sir, nalalabuan pa rin kami. Ano ang sense na malaman namin yung table na yan? So dito tayo sa example. Magbibigay ako ng example. Teka, nawala yung aking view ng Zoom. Dali ah. Mhm. Yan. Nakita nyo na ulit? Sorry, nawala. Nawala sa akin. Okay na ba? Yung blank slide, blank dapat siya. Okay, good. Now, sir, hindi pa rin namin maintindihan anong ibig nyo pong sabihin ng mga suspects. Guys, because of the suspects, there could be drug-drug, drug food, o kaya drug supplement interaction. For you to understand that, let's give an example or let's give some examples. Okay? So proceed tayo sa mga examples for your better understanding. I repeat, related ito sa mga drug interactions. In Tagalog, pag sinabi nating drug interactions, ang gamot kapag ininom mo, again, ulitin ko, if you took a drug, the drug that you took could interact with other components of your body or other drugs that you took. Okay? Pwede siya makipaglaro, makipag-interact. Kaya sa drug interaction, lagi niyong tatandaan na may dalawang characters involved. Meron tayong suspect at meron tayong victim. Okay? Meron tayong suspect at meron tayong victim. When we say suspect, this is the cause of the interaction. Okay? This is the cause of the interaction. And then the victim is the one affected. Okay? The one that is affected in a certain drug interaction. So suspect and also victim. So pag sinabi natin suspect, ito rin yung tinatawag na precipitant drug. Okay? Ito rin yung tinatawag na precipitant drug. And then kapag victim naman, inter or object rather, or the object drug. I repeat, kapag suspect, it's the precipitant, and then kapag victim naman, siya yung object. So far, nagets niyo yung dalawang characters, quote and unquote characters sa drug interaction? Hmm? Okay. So start tayo. Isang object lang ang gamitin natin. Okay? Isang object lang ang ilet nating example. Halimbawa, ang object drug is warfarin. Again, let's have warfarin. Warfarin is highly participative in different drug interactions. So for you to understand enzyme induction and enzyme inhibition, let's pair warfarin with two suspects. Okay? I-pair natin siya sa dalawang suspects. So the first suspect that we will match with warfarin, halimbawa rifampin. Halimbawa lang, rifampin or rifampicin. Again, rifampin or rifampicin. Recall, on your table, ano ang rifampin being a suspect? Is it an inducer or inhibitor? Very good. So ang rifampin is an enzyme inducer. Pag sinabi nating enzyme inducer, rifampin can stimulate the enzyme. It can stimulate the enzyme that would metabolize warfarin, that would inactivate, that would degrade, that would destroy warfarin. So with that being said, ano ang mangyayari sa metabolism ni warfarin? Enhance or reduce? Answer. What will happen to metabolism of warfarin? Enhance or reduce? Metabolism. Metabolism. Sino? Oh, okay, ulit. Makinig ha, makinig. Makinig mabuti. Wala mo nang sasagot. Makinig sa akin. Ang metabolism, bakit nagkakaroon ng metabolism? Sino ang nag-a-act? 'Di ba mga enzymes, mga enzymes. Nung uminom daw ang pasyente ng warfarin kasama ng rifampin, ang ginawa ng rifampin, pinarami niya, nakikinig kayo, pinarami niya ang enzyme na sisira na magme-metabolize kay warfarin. So question, how was the rate and extent of metabolism involving warfarin? Kumusta ang warfarin in terms of metabolism? Na-enhance mo ba ang metabolism niya o na-reduce mo ang metabolism? Very good. Enhanced. Enhanced. Nagets nyo? Nagets nyo ba? Okay, good. Susunod, hindi pa tayo tapos. Enhanced metabolism of warfarin. Now, pag sinabi nating enhanced metabolism, marami o konti ang inactive form niya? Answer. Come again. Marami or konti? Marami. Maraming inactive. Question, pag maraming inactive, less or more effect? Less or more effect brought about by warfarin? Less. Very good. Less. Kaya taken all together, makinig ang warfarin ay isang gamot that can cause bleeding. Di ba na-discuss natin yan last time? Warfarin causes bleeding. Yan yung expected effect natin when we use warfarin. However, if warfarin is paired with rifampin at na-reduce ang effect, what could happen? Bleed or clot using warfarin? Clot, very good. Ang tawag doon, thrombosis. Kaya ang effect niya, thrombosis or clot. Okay? So nagkakaroon ng thrombosis or clot. Overall, nagets ba ang first example sa drug interaction? Nagets? Very good. So check tayo sa unang ka-pair ni warfarin. Next, punta tayo sa pangalawa. Ready na ba sa pangalawa? Ready? Okay. Can you give me an example enzyme inhibitor sa table? Hindi ko na ma-open yung ebook ko eh. Ano ba ang magandang enzyme inhibitor? Okay. Nauna na ako sa inyo. Sige. Cimetidine. Nakita niyo yung cimetidine dyan? Very good. So cimetidine. Ang cimetidine, anong klaseng suspect siya? Inducer or inhibitor? Obviously, it's inhibitor. Imagine, a patient took warfarin together with cimetidine. What does cimetidine do? Does it enhance or reduce enzymic action? Does it enhance or reduce enzymic action? Reduce enzymic action. Pag reduce ang enzymic action, how's the process of inactivating warfarin? Reduce or enhance? Reduce. Very good. So reduce metabolism of warfarin. Pag reduce warfarin's metabolism, do you think na e-enhance ang kanyang pharmacological action? Na-enhance ba yung effect niya? Hindi kasi siya na may metabolize. So extended ba yung action niya? Yes or no? Yes. Kaya extended ang kanyang action, enhancing its effect. However, if this is continuous, pag tuloy-tuloy siya, ang tendency, hindi natin ma-inactivate ang warfarin. So, na-i-intensify ang kanyang effect, kaya pwede magkaroon ng extensive bleeding. Kaya nagkakaroon ng extensive bleeding. So, in enzyme inhibition, na-gets niyo yung bleeding? Hello, nagets niyo yung bleeding? Kung ba't nagbi-bleed? Nawala ba ako? Okay, so these are the possible consequences ng drug interaction. Okay, ano napansin natin? Iisang object, iisang biktima, pero dalawa ang possible effects depending on the suspect. So overall, ang takeaway nyo dyan is, if you know the examples of enzyme inducers and enzyme inhibitors, you can predict if the object's effect would be enhanced o kaya reduce. Kaya sa mga tasks o kaya sa exam na pwede nating ma-encounter, maaaring magbigay ako ng case study doon na merong naka-mention ng mga drugs, and then you can predict the metabolism of the object kung enhance ba yon o reduce. Kaya I highly recommend that you memorize the different enzyme inducers and inhibitors, and also kung ano yung kaya nilang gawin as suspects or as precipitant drugs. Did you get that? Type done if you're finished copying this slide, tapos proceed tayo sa next portion. Good. By the way, bago ko mag-proceed sa next part, yung in-example ko kanina, drug. Meron din tayong drug food. and then drug supplement interaction. Pag binasa nyo yung page 49, may mga examples dyan na mga food o kaya mga supplements that could either be an enzyme inducer or enzyme inhibitor. So pag nagkaroon tayo ng task, for example, pwede ko namang specify. Halimbawa lang, wala sa book, pero may nailagay akong ibang compound, ibang herbal supplement, ibang food, halimbawa. Kung sakaling wala sa notes nyo, pwede ko namang specify doon kung inducer siya or inhibitor. Pero pag nasa book, kasi may libro din ako dito, hindi ko sasabihin yun, kaya you have to memorize or i-check nyo mabuti kapag nagsagot kayo ng task. Anyway, let's continue. Proceed tayo guys sa different metabolic pathways and categories of drug metabolism. So ito na yung medyo malalim na part, ay palalalimin na natin siya lalo. So regarding the pathways of drug metabolism, in general, as I've mentioned numerous times, ang metabolism, ang goal is to make the drug inactive. Pag sinabing inactive, the drugs are not capable already of inducing their pharmacological actions. That's why in other references, aside from the term biotransformation, ang tawag din sa metabolism ay detoxification or detoxication. Because when we lose the pharmacological activity of a drug, we make them inactive and non-toxic. Yun yung general rule, a general rule. However, I think it was mentioned before that not all the time ang product ng metabolism ay non-toxic o kaya inactive, right? Meron tayong cases wherein nababioactivate siya because of chemical conversion. For example, we have acetanilide. Ang acetanilide ay isang compound, isang drug na kapag nag-undergo ng hydroxylation, once it undergoes hydroxylation reaction, it becomes more active analgesic and antipyretic agent called acetaminophen. So kapag na-hydroxylate siya, meron tayong acetaminophen. And then, parathione. Ang parathione, ang route of entry niyan sa katawan natin or yung pagpasok niya sa katawan natin is through inhalation. After inhalation of parathione, this undergoes desulfurization or it becomes desulfurized. It becomes desulfurized, resulting in the formation of paraoxone. Ang paraoxone is highly toxic. This is highly toxic metabolite that can cause extensive parasympathetic toxicities. Again, we have parathione. Through inhalation of parathione, it may be or it will be desulfurized, resulting to paraoxone formation that is highly toxic. Deadly ang paraoxone because it intensifies parasympathetic responses. Kaya pwede siya mag-cause ng death sa patient. So yan po yung mga sample exceptions to the rule that could bioactivate the drug. Bottom line, it's not always losing the action. It's not always formation of non-toxic metabolites. There are cases na kabaliktaran o hindi nagmamatch sa general objective. Okay? And also, hindi ko nasulat dito, pero pro-drugs kasali rin, di ba? Na-discuss natin siya before. Are you still good? Ha? Nakakaintindi pa ba tayo? Hello? May lag ba sa sa akin? Nawala ba ako? Or sa chat? May delays ba sa chat? Sorry. Hi. Feeling ko may delay. Or hindi lang kayo nasagot. Hello? Nandiyan pa ba kayo? Ha? Ayun. So hindi natin malalaman kung may problema ako or wala kapag hindi kayo nasagot. So good. Next, also it was mentioned that the liver is the primary site of drug metabolism. Basically kasi it contains high concentration of enzymes. So kapag maraming enzymes, mataas ang chance na mangyari ang metabolism. That's why it is the major metabolizing organ. However, it was introduced also earlier that hindi exclusive sa liver ang site of drug metabolism because there are other organs and tissues that contain metabolizing enzyme, such as nervous tissues, gastrointestinal tract, kidneys, lungs, and also plasma. So with this, let me give you some cases or examples para mas ma-visualize nyo yung ating sinasabi. Let us start with the nervous tissues. When we say nervous tissues, ito yung ating nerves, mga neurons natin. Ang mga neurons natin, they also possess metabolizing enzymes for drugs. Like for example, monoamine oxidase. Halimbawa, we have monoamine oxidase. Ang monoamine oxidase ay makikita natin sa mga nerve tissues, and monoamine oxidase is responsible for metabolism of biogenic amines or catecholamines. For instance, we have metabolism of norepinephrine, we have metabolism of norepinephrine, also metabolism of epinephrine, and also metabolism of dopamine. Again, monoamine oxidase can metabolize norepinephrine, epinephrine, and also dopamine. That would make them inactive, syempre. Okay? That would make them inactive. So yan ang function ng monoamine oxidase sa ating nerve tissues. So mas maiintindihan nyo yan when you have your pharmacology already. So I'm just stating some enzymes action in this case. And also we have another acetylcholinesterase enzyme. I repeat, number 2 example under nervous tissue, we have acetylcholinesterase enzyme that can cause metabolism of obviously acetylcholine. Again, we have monoamine oxidase for norepi, epinephrine, dopamine, and then acetylcholinesterase naman, metabolism of acetylcholine. So pag na-metabolize ang acetylcholine, inactive din siya. So magkakaroon ng inactive products. So yan ang function ng mga enzymes in the nervous tissue with their corresponding target substrates. You can type done if you are finished copying. So antayin ko kayo and then proceed tayo sa second. Okay, done. Next, proceed tayo sa second. Sa gastrointestinal tract din natin, merong mga metabolizing enzymes. Magbigay tayo ng example na related din sa drugs or sa gamot. So for instance, we have peripheral dopa decarboxylase. Again, for example, we have the enzyme peripheral dopa decarboxylase. Again, in the GI tract, we have the enzyme peripheral dopa decarboxylase. What's the function of peripheral dopa decarboxylase? This enzyme is responsible in the metabolism of the drug called levodopa. Again, this is responsible in the metabolism of the compound called levodopa. Now, kapag na-convert or pag na-metabolize ang levodopa via this enzyme, ang product na nabubuo, dopamine. Ang product na nabubuo ay dopamine. So let me repeat, we have peripheral dopa decarboxylase. Via this enzyme, we tend to metabolize levodopa, forming now dopamine. Now, in clinical practice, paano nyo siya ma-appreciate? Anong importance nung pag-intindi natin ng peripheral dopa decarboxylase? So para maintindihan yan, let me explain the concept of Parkinsonism as a disease and treatment. So discuss natin ang Parkinsonism. Familiar ba kayo sa Parkinsonism? Very good. In Parkinsonism, just to explain briefly, in Parkinsonism, this is a central or CNS disease characterized by low levels of dopamine. Again, ang Parkinsonism, this is characterized by low levels of dopamine in the body. That's why ang goal natin, I repeat kung nakikinig pa kayo, ang goal natin para ma-manage ang Parkinson's is to increase dopamine levels. Again, that is to increase dopamine levels. Yan ang ating goal in the treatment of metabolism. Guys, are you still there? Pwede natin continue? Very good. So I repeat, the goal is to increase dopamine levels. Now, ang una nating maiisip, makinig mabuti ha, hypothetically speaking, ang una nyong naiisip para maggamot ang Parkinson's ng tao, mag-administer ng dopamine. So at first, ang initial thinking nyo, susulat ko yung initial thinking para mas maintindihan nyo. That is to administer. So iisipin natin to administer dopamine in the body para mapataas siya. By the way, bago ko makalimutan, sa Parkinson's, mababa ang dopamine sa utak mismo. Sa utak siya mababa. Sa utak. Hindi yung literal na katawan natin, but in the brain only. So ang goal is paramihin ang dopamine sa utak din. Kaya dapat ang goal natin is to administer dopamine sa utak makarating. So kung i-imagine niyo, alangan namang i-inject ang dopamine sa utak, ang weird nun, ang hirap nun, di ba? Kaya since imposible yung pag-inject ng dopamine diretso sa utak, ang maiisip natin dito initially is to administer by mouth. I-administer siya by mouth. Now, ang problema kapag in-administer siya by mouth, nasusundan? Okay. Ang problema pag in-administer siya by mouth, ang dopamine is not permeable. This is not permeable in the triple B. So ang problem sa dopamine, hindi siya permeable in the blood-brain barrier. Kaya pag binigay mo ang dopamine mismo by mouth, wala rin makakarating sa utak. It won't treat Parkinson's at all. Kaya problema siya. Kaya ang remedy natin dito, instead of administering dopamine itself by mouth, kino-convert or pin-repair ang dopamine para maging levodopa. Kaya ang gamot for Parkinson's na ina-administer by mouth is levodopa. Kasi ang levodopa ay permeable sa brain. Kasi ang levodopa ay permeable sa blood-brain barrier. So permeable siya sa blood-brain barrier. And pwede ko na ituloy. Okay. So ang levodopa, ang kagandahan sa kanya, permeable siya sa blood-brain barrier. So when the time comes na nakatawid na siya sa blood-brain barrier, ang levodopa, mamimit niya ang enzyme na dopa decarboxylase. Okay? Mamimit niya ang enzyme na dopa decarboxylase. That's why pagpasok niya sa brain, ang tendency, mako-convert siya into dopamine. Kaya question, sa dulo ba nakarating sa utak ang dopamine? Yes. So yan ang reason kung bakit natin pine-prepare ang dopamine as levodopa para makatawid siya sa ating utak. Type done if you're finished copying. Hindi pa yan tapos, may susunod pa ako i-explain. Done? So continue natin. Okay. Focus tayo everybody kay levodopa. Again, focus tayo kay levodopa. Ang mga properties ni levodopa are the following. Number 1, ang levodopa ay isang gamot na ina-administer by mouth. Okay? Ina-administer siya by mouth. So kung ina-administer siya by mouth, ang daan niya GI tract din. GI tract. Now, the reason why I am telling you this kasi kapag tinake ang levodopa sa ating by mouth at dumaan siya sa ating GI tract, meron siyang problemang ma-encounter. And what is it? Ano siya? Pag tinake mo siya by mouth, remember, sa ating GI tract, nakikinig pa ba? Sa GI tract natin, merong peripheral dopa decarboxylase. Recall. Anong ginagawa ng dopa decarboxylase? Anong ginagawa niya? Sinisira niya ba ang levodopa? Or ginagawa niya bang dopamine ang levodopa? Yes or no? Yes or no? Yes, very good. Kaya ang tendency, pag dumaan sa GI tract ang levodopa at naging dopamine siya because of dopa decarboxylase, ang realization nyo dyan, sa GI tract pa lang, dopamine na siya. Dopamine na siya. Question, pag prematurely naging dopamine ang levodopa, tatawid ng triple B? Yes or no? No. So pag hindi nakapasok sa triple B, nakarating sa utak, yes or no? No. Very good. No. Kaya ang take away nyo diyan, oo, ginagawang levodopa ang dopamine, pero levodopa itself is a problem already. Problema pa rin siya. Kasi nagiging dopamine siya prematurely. Kaya anong goal natin? O ito yung ultimate star. Ito na talaga yung final dito. Final input. Final input. Pag sinabing star remedy, okay, remedy. Ang star natin dito or ang remedy is to combine with carbidopa. Okay, combine with carbidopa. That's why sa drug for Parkinson's, ang levodopa ay kino-combine with carbidopa. So right now, i-discuss natin ang carbidopa. Ang carbidopa ay isang peripheral dopa decarboxylase inhibitor. Again, carbidopa is a peripheral dopa decarboxylase inhibitor. Please type done if you are finished copying so far bago natin i-explain. Done? Okay, nice. So this is being combined with carbidopa. O makinig guys, ang levodopa para maiwasan ang premature metabolism, kino-combine with carbidopa. Ang carbidopa ay peripheral dopa decarboxylase inhibitor. It means that ang carbidopa, pinoprotektahan niya ang levodopa para hindi maging dopamine right away. Nagets nyo? Nagets nyo? Okay. And when the time comes, again, when the time comes na makarating ang levodopa at carbidopa sa blood-brain barrier, carbidopa cannot enter the triple B. It does not enter triple B. Only levodopa crosses the triple B. Kaya pagdating sa utak, sino na lang ang natira sa loob ng utak? Si levodopa na lang ba ang nasa loob ng utak? Yes or no? Yes. So, ang levodopa, pag nakapasok ng utak, magiging dopamine na ba siya eventually? Yes. Kaya in the clinical practice, wala kayo makikita na levodopa alone available in the market. Levodopa plus carbidopa lagi ang drug preparation. Okay, that's the rationale. Ang dami ko sinabi, no? Pero yan ang rationale. But kidding aside, naintindihan niyo yung dopa decarboxylase and relationship sa mga gamot na diniscuss natin? Okay, nice. Yung slide na to tatapusin ko lang muna bago tayo mag-health break. Pero just to check if you really understood peripheral dopa decarboxylase, please answer what is being asked. Okay, makinig sa akin, pakisagot yung mga itatanong ko. Burahin ko na to just to check. Okay, number 1, peripheral dopa decarboxylase. Saan makikita ang peripheral dopa decarboxylase? Peripheral ha, sa utak o sa GI tract? GI tract. Pag central dopa, sa utak yon. Yun yung difference. Okay? Next, is dopamine permeable through the triple B? Yes or no? No. Is levodopa permeable? Yes or no? Is carbidopa permeable? Yes or no? Sino ang choppy yung huling answer? Is carbidopa permeable sa blood-brain barrier? No. Okay, the answer is no. Next, sino yung peripheral dopa decarboxylase inhibitor? Levodopa or carbidopa? When levodopa is, makinig, true or false yung next mong question, when levodopa is administered alone, it undergoes premature metabolism, yes or no? Sorry, true or false? True or false? True. Okay, true or yes. That's right. Yon. At least naintindihan na yung sa GI tract. Are you learning so far? Okay, continue natin. Hindi pa po tapos. Sa kidneys tayo. Ay, kidneys. Meron din tayong enzymes na makikita sa kidneys na na important rather na important sa clinical practice. So that is the enzyme renal dehydropeptidase. Again, that is the enzyme renal dehydropeptidase. So anong klaseng enzyme ang renal dehydropeptidase in terms of drug metabolism? Renal dehydropeptidase is responsible for the metabolism of the drug called imipenem. Again, that is metabolism of the drug called imipenem. Ang imipenem ay isang antibacterial agent. Madidiscuss natin ito sa final grading period, but para may idea kayo, explain ko na briefly right now. So this is responsible for the metabolism of imipenem, which is an antibacterial agent. Now, what's the implication if this agent was metabolized by the enzyme? Ang effect nito, guys, is of course decreased activity of imipenem that can cause decreased activity, decreased antibacterial effect of imipenem, and in some cases, pwede siya mag-cause ng toxicity. Okay? It can enhance the risk of toxicity. So decrease activity, decrease antibacterial activity, and enhance toxicity. So with this being said, question, problema ba ni imipenem ang dehydropeptidase? Yes or no? Yes, very good. Kaya in the clinical practice, again, in the clinical practice, ang imipenem is being combined with the drug called cilastatin. This is being combined with the drug cilastatin. What's with cilastatin? Ang cilastatin is an inhibitor of renal dehydropeptidase. Again, this is an inhibitor of the enzyme renal dehydropeptidase. Question, guys. Kapag ang cilastatin ay binlock ang renal dehydropeptidase, do you think these events could be prevented? Yes or no? Yes, very good. Yes. Kaya in the clinical setting, ang commercially available imipenem may kahalong cilastatin lagi. Kaya makikita nyo sa generic name, if ever ha, pag nag-internship kayo or kung saan nyo man ma-encounter, makikita nyo lagi imipenem cilastatin and then brand name and then brand name. Kung ano mang brand siya. Hindi ako familiar sa brand eh, pero imipenem cilastatin. Gets? Ha? Okay na tayo dito? All good? Hmm? Okay. So since it's almost 8:30, so health break muna tayo. After the break, proceed tayo sa mga phases of drug metabolism na. Honestly speaking, naiintindihan yung dinidiscuss? Hmm? Okay. By the way, bago tayo mag-health break, sige, brief review tayo para masundan yung dinidiscuss natin. O sa contents na lang. O alalahanin nyo ha, sa contents, yung introduction, define natin ang metabolism, yung importance, drug safety, factors, genetic, physiologic, kinetic, dynamic, environmental, drug interactions. That is, yung mga enzyme induction and enzyme inhibition. Sa metabolic pathways, diniscuss ko dito na liver is an important organ for metabolism. However, there are extrahepatic tissues that contain enzymes, such as nervous tissue, GI tract, kidneys. Yung lungs pala tsaka plasma nalimot ko, pero mabilis lang yon. Sulat ko lang na mabilisan. Balik tayo sa part na to. Okay? Sa lungs at sa plasma, cytochromes ang makikita. Cytochromes. Mamaya, madidiscuss natin ang cytochromes. O sulat nyo lang, cytochromes. Bale, yun ang na-discuss natin so far. Are we good? Pwede tayo mag-health break na? Okay na? Pwede na? Sige, balik tayo na. All right, guys. Moving on to our discussion on drug metabolism, we are now on the different categories of drug metabolism. Accordingly, there are two categories, and there are certain chemical reactions that are categorized in each phase or category. I-discuss natin. Phase 1 reactions include oxidation, including hydroxylation, reduction, and also hydrolysis, in which the dominant chemical reaction under phase 1, we have oxidation. Later, magfo-focus tayo sa oxidation. And then, the major goal of phase 1 drug metabolism, that is to introduce, to modify, or to remove functional group. So the point of introducing a new functional group to the drug, magdadagdag daw ng functional group, o kaya the purpose of changing the existing functional group, or di kaya the purpose of removing an existing functional group is to be able to enhance the polarity of the substrate. When I talk of substrate, the drug intended to be metabolized. Again, let's repeat. The purpose of phase 1 reaction, via these chemical reactions, we can either add a new functional group, we can modify an existing group, or we can remove a certain functional group to be able to enhance the water solubility or the polarity of the substrate or the drug intended to be metabolized. That's the purpose of phase 1 metabolism. In some cases, the goal of phase 1 metabolism is to make the drug qualify to enter the next phase, and that is called phase 2 reaction. Ang phase 2 reaction or conjugation naman, mamaya madidiscuss din natin siya. Phase 2 reaction, the goal here is to add only. Okay, to add. That is to add a new functional group. So how can we differentiate phase 2 from phase 1? Ang phase 1 mapaglaro siya kasi we can add, we can modify, or we can remove. But ang phase 2 is strictly for addition of new group only. Did you get the difference? Okay. So regarding addition of functional group or conjugate kung tawagin, ang tawag sa group na dinadagdag dito ay conjugates. Kaya conjugation ang tawag sa reaction. So this could be acetyl functional group, it can be a sulfate, it can be glucuronic acid, or certain amino acids. So madidiscuss natin ito individually as we go along through our discussion. In phase 2 reaction, just like phase 1, we are further intensifying the polarity of the synobiotic and the importance of making it more polar, that is to qualify the drug eventually for excretion process. Note class that most substances undergo both phase 1 and phase 2 reactions sequentially. So yan yung mga categories of drug metabolism. Now, as I've mentioned a while ago, we will discuss the different chemical reactions under each phase. Kaya ang mga succeeding portions ng ating PowerPoint ay naka-dedicate sa bawat phase of drug metabolism. Starting off with phase 1, drug metabolism. Under phase 1, this is also known as functionalization phase. Again, this is also known as functionalization. What is the reason why it is termed as such? The reason kung bakit ang tawag sa kanya ay functionalization, kasi nga pinaglalaroan niya yung substrate, di ba? Fina-functionalize niya. Pwede niyang i-modify, pwede niyang dagdagan, o pwede niyang alisan. Depende sa sitwasyon. Just to make the drug polar. So that's phase 1 reaction. And remember, we have 3 major reactions involved in phase 1. These are oxidation. So kasali dito yung kanina na hydroxylation. Kaya ako sinabing including hydroxylation. And also we have hydrolysis and reduction. So ito yung mga chemical reactions. Recall question, amongst the 3, sino ang dominant mechanism of phase 1? Very good. That is oxidation. So that's the dominant phase 1 metabolism. With that being said, let's proceed to oxidation reaction. As I've said, oxidation is considered as the most common reaction involving synobiotic metabolism, particularly in phase 1. Now, ang oxidation ay nangyayari, makinig, ang oxidation ay nangyayari because of enzymes. Okay? Because of enzymes. Now, enzymes capable of oxidation, again, enzymes capable of oxidation can vary depending on their source or yung kanilang properties. Because of this principle, there are two mechanisms of oxidation. Again, I repeat, may dalawa tayong mechanisms of oxidation because iba-iba yung mga characteristics na mga oxidizing enzymes or oxidative enzymes. These are the following. Number one, we have cytochrome-mediated oxidation. And then second, we have non-cytochrome-mediated. Let's start with cytochrome-mediated. Kaya siya tinawag na cytochrome-mediated kasi yung cytochrome itself, enzyme siya. Okay? Enzyme yung cytochrome. So cytochrome ang tawag sa enzyme na nag-oxidize. Kaya cytochrome-mediated ang tawag. Mind you that majority of oxidative reactions are caused by cytochromes. Kaya ito ang dominant mechanism of oxidation. Okay? Ito yung dominant mechanism of oxidation. In fact, more than 50% of commercially available drugs ay nag-undergo ng CYP-mediated oxidation. Proceed tayo sa non-CYP. Yes or no? Yes? Good. Next, what about non-CYP mediated? Non-CYP basically means that the enzymes capable of oxidation are not members of the cytochrome family. Ay, super family kasi ang cytochrome, marami sila. Now, kapag ang enzyme na nag-oxidize ng gamot, hindi siya characteristic ng cytochrome, common sense, non-CYP ang tawag. So yan yung dalawang mechanisms of oxidation. So ito yung ifo-focus natin right now. Starting with cytochrome-mediated oxidation. As I've said, ito ang dominant oxidative reaction involving medications because of cytochrome. Now, ang enzymes that can oxidize are called cytochrome. Kaya at this point, ipaliwanag natin kung ano ba yung mga enzymes na cytochrome. Kaya yung nakabox ngayon ng color pink, yan yung description natin about cytochrome. What do we mean by cytochrome? Cytochrome or CYP stands for cytochrome P450 monooxygenase enzyme system. So as you can tell, ang haba ng pangalan kasi super family siya. So for our convenience, ang itatawag na lang natin sa kanya, ang itatawag na lang natin sa kanya ay CYP450 or CYP450 for our convenience. So CYP450 is a group of membrane-bound monooxygenases that are found in the smooth ER and in the liver as well as in the extrahepatic tissues. So this is also known as mixed function oxidase or microsomal hydroxylase. So yan ang mga CYP superfamily or CYP450. The main function of CYP450 is to provide electron transport system. Okay? That is a multi-component electron transport system that would eventually lead to oxidation of substrate. Okay? That would eventually lead to oxidation of substrates. So please stay tuned sa term na electron transport system kasi mamaya pag diniscuss ko yung catalytic cycle, mamaya, dun yung makikita na merong transfer of electrons involving CYP450. So medyo chemical na yung discussion natin. Pero syempre, uunti-untiin natin para maladerize yung inyong understanding. Okay? O review muna tayo. Ang CYP450 ay superfamily na ang goal ay magpasa-pasa ng electrons para eventually ma-oxidize ang gamot o ang substrate. Can you follow so far? Okay, next. Meron kayo nito? Meron kayong slide na to? Very good. So discuss natin ngayon ang components ng cytochrome P450. The CYP450 or CYP450 family has three major components. These are number one, the heme protein, Second, the flavoprotein. And then finally, phospholipid. Take note, protein components ang heme at flavoprotein. These are protein components. And then, apparently, yung huli, hindi siya protein. Lipid siya, ay non-protein component. Phospholipid, that is in the name of phosphatidylcholine. Now, for the heme protein, for our convenience also, itatawag natin ang heme protein as P450. Okay, again, for our convenience, ang heme is P450. Different siya sa CYP450 kasi in totality ang CYP450. Ang P450, the heme protein itself. So first, we have the heme protein. Heme protein is very important because it is considered as the central component in the functioning of the whole CYP450 family. So mamaya papakita ko ang structure ng heme protein and its importance. Next, flavoprotein and phospholipid naman, again, flavoprotein and also phospholipid, important naman sila for electron transfer. Ay important naman ang flavoprotein and also phospholipid for electron transfer. So for instance, for flavoprotein, it facilitates electron transfer by the action or by the assistance of the enzyme NADPH-P450 reductase. Okay? And then ang phospholipid naman ay source din ng electron for transfer, and it will be transferred also by the same enzyme, that is the reductase as well. So these are the components of P450. So recall natin, pinaka-importante ang heme, and then flavo and phosphatidylcholine for electron transfer. So yan ang components ng superfamily. So mamaya madidiscuss natin ang mga specific roles nila. Next, pwede na po dito sa figure. Okay, makinig mabuti. So let's proceed on the most important or the central component of the cytochrome superfamily. I repeat, sa tatlo, we have heme, flavo, and we have phospholipid. Sa tatlo, ang pinaka-importante ay ang heme protein. So pag-aralan natin kung ano ang itsura ng heme protein P450. Number one, ang ibig sabihin ng heme, makinig, ang ibig sabihin ng heme, narinig nyo na to sa biochem last time, heme? Very good. So i-recall natin kasi related talaga siya. Pag sinabi nating heme protein or heme itself, heme is an example of a protein containing central iron ring structure. Okay, meron siyang iron, ay iron atom or iron metal at the center. And this is attached with protoporphyrin. So meron siyang protoporphyrin. So heme is a protein containing iron with the so-called protoporphyrin. Ang protoporphyrin, siya yung napakalaking ring structure na naka-shield sa iron. Okay? Ito yon, yung malaki na yan. Yan yung ring na protoporphyrin. Now, the reason why iron is linked with protoporphyrin, that is because meron siyang links attached. Meron siyang mga bonds attached. So sa iron, take note, meron tayong 6 na coordinated bonds sa iron. So we have 6 coordinated bonds sa iron in which the first 4, 1, 2, 3, and 4, ito yung mga nakadikit sa nitrogen. Ay, ito yung nakadikit sa nitrogen. And these 4 coordinated bonds are the ones designated for the attachment of iron with the ring itself. Ay, with the ring itself. Ulitin ko ha, ulitin ko. 6 ang coordinated bonds ng iron sa heme protein at 4 doon ang nakadesignate para dumikit yung iron sa gitna with the ring protoporphyrin. Na-gets yung 4 na coordinated bonds? Hello? Na-gets? Okay, that's why if you will refer on the figure, makinig, if you refer on the figure, ito yung central iron, this is the central iron, and there are 1, 2, 3, and 4. Lahat ito ang nakadesignate para dumikit yung ring na malaki na protoporphyrin sa iron sa gitna. Okay? Next, punta tayo sa fifth at sa sixth coordinated bond. Again, punta tayo sa fifth at sa sixth coordinated bond. Dito tayo sa panlima. Again, panlimang coordinated bond. Ang tawag natin sa panlimang coordinated bond is the proximal, ay proximal bond. Ay proximal coordinated bond. And then yung pang-anim naman, ay ang distal, ay distal coordinated bond. So I repeat, panlima is the proximal and pang-anim ang distal coordinated bond. Now, very important in understanding P450, a P450, that is the state of iron, the charge of iron at the center. Sa P450, ulitin ko, sa P450, unlike hemoglobin, ang P450, ang state ng iron ay 3 positive. Pag sinabing 3 positive, ano to? Ferrous or ferric? Ferric, very good. So ang state niya ay ferric. So anong importance na ferric siya? May kinalaman siya sa fifth at sa sixth coordinated bond. O makinig, kapag fifth coordinated bond, it is attached, again, this is attached with the amino acid residue cysteine. Okay, this is attached with the amino acid residue cysteine. Now, the importance of its ferric state, that is for the substrate to bind, that is for the substrate to bind to it, substrate to bind to it. Pag sinabi nating substrate, ito yung drug na intended to be oxidized. That is the drug intended to be oxidized. So cysteine, component ng fifth coordinated bond ang cysteine, in which important eventually ang pagdikit ng gamot sa mismong P450 para makadikit siya. And also, yung fifth coordinated bond, again, yung fifth coordinated bond, in its ferric state, yung iron sa ferric state, this is also essential, again, this is also essential in the formation of oxenoid intermediate. Okay, this is important for oxenoid intermediate, intermediate formation. Important for oxenoid intermediate formation. Later, madidiscuss natin yung oxenoid intermediate. Madidiscuss natin yan. Stay tuned muna. Sulat nyo lang muna. Pakitype ang done kung tapos ng kumopya. So fifth coordinated bond, and then substrate also, important for oxenoid intermediate formation. And then punta tayo sa sixth coordinated bond. Again, sixth coordinated bond. Makinig guys, sa sixth coordinated bond, it depends sa state ng iron kung anong didikit sa sixth coordinated bond. Again, it depends. It depends. If the charge of iron is ferric, again, kapag ang charge ng iron ay ferric, ang didikit sa kanya water. Ang didikit sa kanya ay water. Whereas kapag ang charge niya ay ferrous or positive 2, pwedeng molecular oxygen, it can be molecular oxygen or CO or carbon monoxide. I repeat, pag ferric, it's water. And then kapag naman ferrous, that is oxygen or carbon monoxide. So yan po ang mga dedicated or designated coordinated bonds na dumidikit sa iron. So I repeat, in P450, ferric ang usual state niya. Okay? Yung first 4 bonds is for the protoporphyrin, that is sa ring. And then the fifth, the proximal bond, that is for the amino, it contains or it has cysteine amino acid residue intended to bind with the substrate or the drug, which in turn would cause formation of oxenoid intermediate. And then sa sixth coordinated bond, if the state is ferric, it's water. If the state is ferrous, it could be oxygen and carbon monoxide. Hoy, nandiyan pa kayo? Ha? Okay. Next. So ito, na-discuss, moving on tayo ha. Ito actually, na-discuss ko na siya. Oxygen activation. Again, P450 is important in oxygen activation and substrate binding. Ano pong ibig sabihin ng vital role in oxygen activation? Ibig sabihin nito, P450 is important, that is for binding of oxygen. In other words, that is to make the ferric state at the center to become, anong state dapat para makadikit ang oxygen? Maging ferrous. Very good. Kaya makikita nyo mamaya sa catalytic process, makikita nyo na pag naging ferrous ang state ng iron, didikit ang oxygen. Okay? Didikit ang oxygen. Important siya. And also substrate binding. Saan dumidikit ang substrate? Anong coordinated bond? Panlima or anim? Very good. Sa fifth coordinated bond. Ito ba yung magli-lead sa oxynoid intermediate nung complex? Yes, okay. So didiscuss natin yan. And then the active site of P450 consists of hydrophobic substrate binding domain in which is embedded to an iron protoporphyrin prosthetic group. Question, bakit dumikit ang iron prosthetic group sa protoporphyrin? How many bonds are allotted for it? 4. Very good. Very good. 4. So ang point na ng slide na to, dito naka-designate yung 6 na coordinated bonds ng iron. Yung una hanggang pang-4, yung pang-5 at yung pang-6. Okay? So yan ang P450. So ngayon, i-discuss natin, ito na-discuss ko na rin ito, itong part na to, yung activate molecular oxygen. So dito tayo ngayon sa requirements. So for your participation, kindly proceed on page 51. Go! Doon kayo sa bullet na requires. Paki-type ang ready if you have seen it. Ready? Okay, very good. So in this part on phase 1 metabolism, to be able for cytochrome oxidation to occur, we need the following requirements. Number 1, the heme protein. Question, ang heme protein, siya yung P450, right? P450. CYP450 or P450 is in a resting state when bound to ferric state. So siya ay resting pag ferric and then active kapag ferrous. Bakit natin siya tinawag na active at its ferrous form? Do you think it can accommodate oxygen in that state? Yes or no? Hindi ko alam yung end nyo kung madaling makita yung ebook tsaka ano, sorry ha. Pero I'll just patiently wait for your responses. Very good, it's a yes. Next, NADH or NADPH. Ano ang importance ng second requirement? These are cofactors of CYP450. In short, guys, ito yung electron source. Ito yung electron source. Important ba ang transfer of electrons sa i-discuss natin? Yes, kaya stay tuned. And also hydrogen atom is needed. Hydrogen atom for abstraction process. And then finally, syempre, molecular oxygen. So yun yung mga requirements. So overall, we have 4 requirements. Heme protein, the cofactors or the reducing agents, and then we have hydrogen atom and molecular oxygen. So taken all together, sige, balik tayo sa aking presentation. Okay, balik tayo sa aking presentation. So taken all together, we can summarize the requirements like this. So number 1, makinig number 1, we have RH. Pag sinabing RH, the drug or the substrate itself, when we say substrate, this is the one intended to be oxidized. Okay? Next, do we need molecular oxygen? Yes, we need molecular oxygen. Next, do we need electrons here? Yes, also. Kailangan natin ng electrons. 2 electrons ang kailangan natin. Question, sino magpo-provide ng electrons? Yung mga reducing agents. Sa ebook nyo, ano yung reducing agents dyan? Yung NADH at NADPH. Tama pa ba yung sinusulat ni teacher? Okay, so sila ang magpo-provide ng electrons. And yung mga flavoproteins o kaya yung NADH ang bahala and also the enzyme. Next, do we need hydrogen? Yes. So we need two hydrogen atoms in this case. So in this portion, pag nakumpleto natin ang mga requirements, the substrate, the oxygen, electrons, and also proton, via the cytochrome, we tend to produce hydroxylated substrate. Pansin nyo, may O na siya, na oxidize na siya. So hydroxylated na yung substrate. And then, minor product would be water. A minor product would be water. So overall, pagkatapos ng oxidation, ang makikita nyong product dito is ROH na, or hydroxylated product, signifying that the substrate has been oxidized and the process ends. Ay, so yun yung mga requirements that would lead to oxidation of the substrate. So yan po yung requirements and target product. 2A, nakakasunod pa ba? Kasi dito na tayo sa tougher part. Okay? Dito na tayo sa susunod. Tougher portion. So bago tayo mag-start sa catalytic cycle, heads up, kailangan nyo ma-check yung page, dapat nasa page 51 kayo guys, yung step A, B, C. Have you seen that? Ha? Okay. Sige, para hindi kayo mahirapan, makinig na lang siguro muna sa akin, tsaka natin i-double check yung libro. Ay, makinig muna sa akin, tsaka natin i-double check yung libro. So bago natin simulan yung discussion on catalytic cycle, review questions muna tayo ulit. Review questions. Ano yung pinaka-important component ng CYP 450? Anong pinaka-important na component ng buong enzyme family? That is the heme component. Ano ang AKA ng heme? Is it CYP450 or P450? P450, very good. Anong atom or anong metal ang nasa gitna? Iron. Ano ang resting state ng iron sa P450? 3 or 2? 3. Ano ang active form? 2. Bakit siya active? Sinong tatanggapin niya? Pang-ilang? Okay, oxygen. Ano ang bond allotted for oxygen? 5th or 6th? 6. Pag 5th, anong didikit doon? Substrate. Okay, substrate. So discussan natin. Meron na kayo mga general knowledge about cytochrome P450. Kaya kahit papano, ready na kayo makinig dito sa catalytic cycle. So this is divided into 8 steps. So first, let's study the heme first, yung heme muna itself. As you can see, yung heme, this part, the heme itself is in its ferric state, so it is still inactive, right? This is still inactive. So the first step is attachment of RH. Ano yung RH, guys, sa ating symbol kanina? That is the substrate. Is the substrate the drug intended to be oxidized? Yes or no? Yes. So substrate will attach with the heme. It will attach with the heme at 5th coordinated bond. That's why we can see here na nakadikit na ang substrate dito sa iron. Okay, dito sa iron. So makikita nyo, meron tayong ferric CYP450 substrate complex. Okay, meron tayong ferric P450 substrate complex. So that's it. Question, accommodating na ba for oxygen yung complex na to after step A? Yes or no? Does it accommodate oxygen na? No, not yet. Very good. Next step, pwede na sa step B? Pwede na po ba sa step B? Okay, step B tayo. Sa step B, it involves reduction of iron. Okay, it involves reduction of iron, making it a ferrous one. Okay, making it a ferrous one. So to be able for reduction to happen, we need a reducing agent. So when we say reducing agent, that is the one that would provide electrons. Okay? That would provide electrons. Di ba kapag electron transfer, lalo na pag mag-gain ng electron, it's reduction, right? And ang reducing agent dito is NADPH. Again, ang reducing agent here is NADPH or pwede ring NADH. So that's why there is electron transfer to the complex, kaya naging ferrous state yung complex. Okay? Kaya naging ferrous yung complex. Okay? Ferrous yung complex. Gets yung step B? Next, pag naging ferrous complex na siya, nakabuka na ba yung coordinated, yung, di ba yung 6 coordinated bond? Anong sabi ko? Pag ferric siya, ano ang meron sa 6 coordinated bond? Water lang, water, di ba? Pero pag naging ferrous siya, bukas na ba siya for oxygen? Yes or no? Yes, kaya important ito for molecular oxygen activation and attachment. Kaya ang step C, it involves oxygenation. Madadagdagan ng oxygen, kaya magkakaroon tayo ng formation ng ferrous P450 oxygen. Ang haba ng pangalan pero complex lang din siya. Okay? So we have ferrous P450 oxygen substrate complex. Okay? Kaya makikita nyo, may oxygen nang nakadikit dito. Do you understand me? Ha? Naintindihan? Okay, next, let's continue. Step D tayo. Sa step D, it involves resonance. Resonance rearrangement. Again, it involves resonance rearrangement that would convert ferrous state into ferric. Okay? That would cause formation of ferric P450 superoxide, superoxide substrate complex. So that can cause formation of the ferric P450 superoxide. This is superoxide because of resonance rearrangement substrate complex. So in short, babalik siya into ferric. Kaya hindi na siya tatanggap ulit ng oxygen. Okay? And oxygen becomes superoxide. So that's step D or step number 4. Next, step 5 tayo. Pwede na sa step 5? Done copying? Ha? Next, step 5. Anong mapapansin natin sa step 5 or step E? May electron na pumapasok. Pag may electron na papasok doon sa reaction, anong reaction ang may isip natin? Oxidation or reduction? Pag may papasok, very good. So magkakaroon na naman dito ng reduction reaction, and usually ang sources dito, pwede siyang NADH pa rin, ay NADH or NADPH. So pwede siyang NADH or NADPH, o kaya pwedeng ang source ng electron would be yung isa pang component ng CYP450 family. Ano pa yung isang electron provider ng buong superfamily na hindi natin nababanggit? Sino pa yung isang electron provider, yung nagfa-facilitate ng electron transfer? Bukod sa NADH, sino pa? Yung pangatlong component. Sino yung pangatlong component? Starts with letter P. Yes, phospholipid. Okay, yung phospholipid. So pwedeng source ng electron yung NADH or NADPH o kaya yung phospholipid. Okay? So yan yung pwedeng sources. Okay? Flavoprotein o kaya yung phospholipid. So that's why mapapansin natin, na-reduce ba ulit yung state ng iron? Yes or no? Na-reduce ba ulit? Yes, okay. So na-reduce siya into ferrous, ay na-reduce siya into ferrous. However, this is considered as a fast reaction process. So short-lived lang siya, and it will undergo rearrangement right away. Kaya mapapansin nyo at the end of step E, babalik na naman siya sa ferric state. So kumbaga, dumaan lang siya na mabilis. And then it became ferric ulit. But take note that the superoxide component has been modified already. So instead of superoxide in step E, it became peroxy component na. So it became ferric peroxy substrate complex o kaya ferric P450 peroxy substrate complex. So ang important lang naman dyan yung modification ng oxygen component. Okay? From superoxide, it became peroxy. Next, step F. So punta tayo sa step F. In step F, it involves heterocyclic cleavage. So it involves cleavage, heterocyclic cleavage. And in heterocyclic cleavage, that causes conversion of this complex, a conversion of this complex into the so-called ferryl, a ferryl oxenoid complex. So it causes formation of the ferryl oxenoid complex. So makikita natin in ferryl oxenoid complex, iron is in its positive 4 state, and then we have molecular oxygen or oxygen, and then the substrate itself. So naging ferryl oxenoid complex siya. Question, narinig niyo na ba yung ferryl oxenoid complex kanina? Yung oxenoid intermediate, have you heard that a while ago? Yes, very good. Ang ferryl oxenoid complex is being observed, this is being observed at the 5th coordinated bond, and this is considered as the active oxygen species, or species. So this is considered as the active oxygen species. Bakit siya active oxygen species? Because kapag naging oxenoid state na yung ferryl complex or yung 5th coordinated bond, that would eventually cause formation of the so-called ferryl hydroxide complex. So that can cause formation of the ferryl hydroxide complex. In short, kapag naging oxenoid complex na siya, that would eventually cause hydroxylation. So na-hydroxylate na yung substrate natin. So makikita nyo, nagka-oxygen na siya. Nagka-oxygen na siya. And then eventually, due to the so-called radical recombination, due to the process called radical recombination, ang tendency, ang P450, it becomes inactive ulit. It becomes inactive once more. And then, mahihiwalay na, the substrate would be separated. So ito na yung oxidized substrate. Again, this is now your oxidized substrate. So what can you observe about the oxidized substrate? May O na ba siya? May oxygen na ba siya sa product? So does it have O or oxygen? Yes. Okay? So at the end of the last step, you can see na ang substrate mo ay na-oxidize na. So somehow, babalikan ko to ha, wait lang. Kaya somehow, if you will check this overview, via these reactions, nakuha natin ang product na ROH, which is the oxidized or hydroxylated product of the substrate. So nangyayari yan because of this catalytic cycle. Okay? Type done if you are finished kasi re-reviewin ko siya briefly para ma-reinforce yung diniscuss. Done? Okay. Done. Very good. So balikan natin briefly ha. Ang importante dito kung paano nagbabago ang charge ng iron at nung oxygen. Paano siya nagbabago? Until such time na maging ROH siya. So start tayo ulit sa inactive form ng P450, and then pag nalagay ang substrate sa 5th coordinated bond, we have this complex. And then reduction reaction. Pag reduction, magiging positive 2 yung iron. Pag naging positive 2, pupunta na ang oxygen sa 6th coordinated bond. Next, resonance rearrangement. kaya magiging superoxide ang state ng oxygen, and then ferric state yung iron, and then step E, reduction ulit, kaya nagiging ferrous superoxide complex. Pero what did I tell you? Does this happen fast? Yes. Okay, short-lived lang siya. That's why through rearrangement, it becomes ferric ulit, pero the state of oxygen has been changed into peroxy. This is important because through cleavage reaction, we can get the active oxygen species. Anong tawag natin sa active oxygen species? Starts with letter O, oxenoid. Okay? Oxenoid complex. Because in oxenoid complex, that would finally lead to hydroxide. Sorry, mali spelling ko na hydroxide. I ito. Hydroxide complex. And through radical recombination, makikleave na ang oxidized substrate, and then babalik sa original state na inactive ang P450. And then the cycle continues. Okay, the cycle continues. So yan ang catalytic cycle, steps in catalytic cycle, kung saan nagamit natin ang mga requirements. Okay, nagamit natin yung mga requirements. So ito na yung last slide ko for today para makapagpahinga na kayo. So last slide para lang ma-reinforce yung diniscuss. Ito ulit yon. Ibang picture lang ang nakita nyo but same principle. Ay same principle lang sila. Number 1, nakikinig pa ba guys? Last slide na to para makapagpahinga na kayo. Okay. First, bigyan ko kayo ng mga hints and then you answer what is being asked. Number 1, this is inactive. Okay? Next, substrate at what ligand or at what coordinated bond? Fifth. Next, makikita nyo, may substrate na. Next, ano nakita? NADPH and then reductase. Sila ba ang nag-i-induce or responsible for reduction? Yes. Kaya may electron. What can you expect sa charge ng iron? Naging 2 ba? Yes or no? Yes. Pag naging 2, sino ang pwedeng tanggapin sa 6 bond? Sino? Oxygen. Pero anong sabi ko kanina? Oxygen lang ba ang pwede? Sa 6 bond? Sa introduction, ano pa yung isa? Yes, carbon monoxide. Kaya dito sa kanan may carbon monoxide. Oh. Nakuha nyo ba? Nakuha nyo? Ha? Okay. So guys, kaya pala siya P450 kasi kapag chinek nyo yung absorbance, habang nakadikit yung mga molecular oxygen or carbon monoxide, ang absorbance niya 450 nanometers. Kaya P450 ang tawag. Gets? Next, dito tayo. Next, kapag na-attach ang oxygen sa 6 coordinated bonds, so meron na tayong molecular oxygen. Actually, mas detailed lang yung previous slide. Dito medyo shortcut eh. Hindi na pinakita yung resonance rearrangement. Diretso ano na siya? Reduction na siya. Ay diretso reduction. And then peroxy, and then cleavage. Anong sabi natin kapag cleavage? Doon na makikita ang activated oxygen species, yung oxenoid, doon na lang kayo mag-base sa oxenoid. So diniscuss ko kanina yung +4, yung +4. And then makukuha na yung oxidized substrate or oxidized product. That is, babalik na ulit sa inactive form yung enzyme. Ayan. Mga bata, naiintindihan yung catalytic cycle? Yes. That's why importante ang requirements. We need the substrate, we need molecular oxygen, we need 2 electrons, and also protons, 2. So that's why mangyayari ang oxidation of the substrate into a certain product. Supposedly, ang next part ng discussion natin would be classifications of P450 multigene family, pero didiscuss natin siya bukas na lang. So kahit ano mangyari, tatapusin natin ang remaining topics ng metabolism tomorrow. So I am positive na matatapos natin siya tomorrow kasi mabilis na lang yung mga susunod na mga reactions. So hanggang dyan muna tayo for today.